Our next speaker is Claire Hankenson. And Claire, if you want to pull up your slides.
Sure, no problem.
She is the director of the Campus Animal Resources and the attending veterinarian at Michigan State University. She holds a faculty position as professor of laboratory animal medicine within the Department of Pathobiology and Diagnostic Investigation at the College of Veterinary Medicine. Dr. Hankenson obtained her vet degree from Purdue and completed her graduate work and residency at the University of Washington. And she became a diplomat of the American College of Laboratory Animal Medicine, or ACLAM, in 2002. She's been active on committees in ALAS, served on the executive board for ACLAM, and is an ad hoc specialist with ALAC, and she serves as a member of Priminar's board of directors now. Her research interests include exploration of refinements in laboratory animal practice with mice and rats, and those include genotyping methods, rodent surgery support, thermoregulation issues, and humane endpoints. So Dr. Hankenson is going to talk to us about the evolution of the 3 R's, reflections on art, ethics, and science.
Thank you so much, Christina. All right, it is really wonderful to be here today. I really would like to express my thanks to the conference organizers for this invitation. And I just want to point out that dozens of people before me for dozens of years have been discussing variations of what I will share today, in the hope that we continue to keep our lines of communication open to better understand our own and others' perspectives. So for the overview of my talk today, I'll share background on the intersection of art as a perspective on evaluating the 3 R's. I'd like to review the classic 3 R's for their animal use and welfare and the subsequent supporting guidance that's come out since they were published. We'll also touch on how to analyze animal research ethics with the progressive 3 R's lens and bring in some of the pandemic examples from that. And lastly, I was asked to share updates on the efforts by the NIH to address rigor and reproducibility in science through a dedicated working group in which I have been able to participate. So I definitely do want to say that the views represented today are my own, and they're based upon many years of reflection and discussions with colleagues that are veterinarians, animal care staff, AWIC professionals, committee members, scientists, and family members. So during the pandemic, like many of you, I gained back a lot of time with the loss of other activities. And when the Philadelphia Barnes Art Museum pivoted to online courses, I enrolled in a few lectures, learning about collections. I had never taken or had time to take art history before. So I learned about painters like Seurat and Van Gogh. And I was in the midst of these art classes when I was asked to participate in this conference on the 3 R's. And as I thought about the framework of the 3 R's and sort of juxtaposed it against the ongoing art classes and the frames, the literal frames that I was seeing around artistic masterpieces, the ideas started to merge in my brain a bit. So when analyzing animal research and the harm versus benefit considerations, the viewpoint is widely dispersed depending on whom is the observer. So are you a member of the public? Are you in the media? How about anthropologists or statisticians or veterinary nurses? Each of these stakeholders in animal welfare will come to the 3 R's with their own perception, and no one person can make that determination about the harms and benefits without discussion and understanding of multiple perspectives. So in this country, we are a part of a science community that has the privilege to deliberate on moral issues with the responsibility to keep objective facts and subjective feelings in balance when ethical considerations are involved. We each may see things, as you can see on this slide, in a different frame, depending on the scenario that's presented to us. However, keeping things boxed in that frame, the literal term framework, potentially inhibits our ability to think more broadly about the balance of ethics, welfare, benefits, and science. So this picture represents the French Salon. This came in one of my art classes. This was an event that was held annually in Paris, beginning in the 1660s, arguably the greatest annual art event in the Western world for many decades to come. So using my art analogy, and looking at this picture, we are the audience today. The scientists, the animal care workers, the public, the ethicists, the regulatory overseers. We are this group of people standing together on the ground, looking up at the walls that are plastered with art. the art of science, the art of animal welfare, the art of all the species that we interact with. Our perspective on how and where to look to get the best view depends on where we stand. So this image serves to remind us that the 3 R's are not stagnant. They're not two-dimensional. They demand continuous discussion and evolutionary interpretation, which is what I like to talk about today. So the 3 R's serve as a universal touchstone with respect to the 3 words we've been hearing about: refinement, reduction, and replacement. And they're used by many people in our community. Russell and Birch established these terms as part of a new applied science in 1959. The goal was to improve animal welfare using the application of these terms for a development tool for scientific methods and medical discovery. In all aspects of animal use, the anticipated benefits of scientific inquiry have to be weighed against the potential detriments borne by the animals themselves. And this is that ethical balance that I've already referred to as the harm-benefit analysis. Over time, rigid interpretations of the classic 3 R's in study design, for example, refinement, and statistical analysis, reduction, and in selection of alternatives to live animals or replacement, have somewhat stifled the spirit of their intent and limited scientific progress in production of translatable and reproducible results. However, the classic 3 R's, as they're listed here today, and we've seen some examples and questions that were raised earlier, when we really look at just these direct terms and how they're listed in bullet points, they're used as this kind of ultimate benchmarking tool against which high quality science in both academic, government, and industrial sectors is measured, with an increasing focus on developing alternative approaches to avoid the use of animals. And that's all very valid, but it's not all that the 3 R's represent. And importantly, they were never initially linked by Russell and Birch to alternatives to animal use. So in thinking about the 3 R's, I'd like us to consider the classic view as somewhat of a closed umbrella. Each of the R's is in a tight fold. In this position, there's one hand holding it. That's the one person that's looking at the 3 R's at that time. So as I said before, the challenge here is that the 3 R's do contribute to the evaluation process, but they are not able to be the sole ethical consideration for animal welfare. And to assume that they can be is really expecting too much of the original intent. They further have been misappropriated sometimes in the sense that if a project isn't accomplishing one or more of the R's, then science and the animal research enterprise are failing in some way, which I have to sort of, raise some objections to in this conference. So it's not necessarily an accomplishment for human benefit if we use fewer animals when we actually have more disease and illness, which has been exemplified over the last year. And it's certainly not a failure if we have to look at more than one sex, male and female, which has been brought up in multiple talks this morning. It's not a catastrophe if we don't replace animals simply because we have to have that intact body system and measurable responses for observations, interventions, and outcomes. So again, as we move forward in transitioning then and taking the folded umbrella analogy, let's expand it. Let's allow more room for interpretation of the wording and intent. Let's put more hands underneath the umbrella as stakeholders. And let's talk about concepts that appeared after the 3 R's were published in the 1950s that are important now and continue to synergize with the original 3 R's. For example, in 1965, the 5 freedoms were released. They were designed to address the livestock husbandry industry in the UK. And the classic 3 R's, even though they came out and were presented in '59, they really didn't become common in the vernacular until the 1980s. So as the stakeholders begin to talk about it, and we at this conference, the attendees, the panelists, we are the stakeholders. We should be coming together to rigorously evaluate the science, the proposals, the designs, the literature searches like we just learned about, the animal species, the models we select, and the impacts. So I do want to point out again some of the supporting items that came out after the 3 R's were published. And in particular, this one is still very critical. It's the US government principles, of which there are 9. And I'll show you these here. Now, they're very important. I'm not going to take the time to go through all of them because hopefully everyone is aware of them. They are in the back of the current edition of the Guide for the Care and Use of Laboratory Animals. But they were released in '85 by the federal government and designed to be such that federal agencies would recognize that the new knowledge necessary for improvement of health and well-being, as well as other animals, requires in vivo experimentation with a wide variety of animal species. And so the two that I highlighted here are in gold at the bottom. The second principle is that the procedures should consider relevance to human or animal health, advancement of knowledge, and the good of society. This continues to be reiterated today and will be in the future. And the other really important point was the ninth principle at the top in green. The decisions of these principles should not rest with just one group, not just the investigators, not just the people that come up with the ideas, but also with groups that ethically review things such as the AWIC. And I do just want to pause a moment here on the AWIC because it's worth mentioning that it also came into being as a mandatory review group in 1985. And since then, the AWIC enterprise has really become incredibly robust and specialized as a field. With all of the critical responsibilities of the AWIC and the members' expectation to review protocols, look at facilities, do post-approval monitoring, and respond to concerns, it's really unrealistic that we put so much on this committee that they can be the only ethical arbiter of animal research. And this is still a concern that I hear at different times, that the AWIC should do more and really think about the ethics and improving protocols. So it's really impossible for us to set standards on AWIC deliberations and decisions when the people and the expertise will differ across committees at different sites. However, the good thing is that flexibility was built into that system early on so that we could bring in consultants and subject matter experts. They can always be invited to assist with review discussions. And again, as I've mentioned, just in the last year, external to my institution, I've been in discussions where the AWICs have been blamed for the reproducibility crisis because they're not mandating things like reduction of numbers. For those in the public that may not understand the institutional review process, then I say we need more transparency to clarify exactly how decisions, critical decisions, get reached about approvals for animal studies. This transparency would help the scientists themselves. They may not understand why a protocol is being held up at the AWIC level for more discussion. And then the public will be able to understand potentially better that we do have a very robust review process that considers animal welfare at the center. Jumping ahead to a more recent impact on ethics and scientific design, I wanted to bring up the 4 Cs. This was coined by the NIH in 2016, and we've already touched on this a little bit earlier today, but these are the 4 Cs to consider, collect, characterize, and communicate. So women now account for roughly half of all participants in NIH-supported clinical research. However, more often than not, basic and preclinical biomedical research has focused on male animals and cells derived from males. So this over-reliance on male animals may obscure the critical key sex influences on health processes and outcomes. So NIH has expected that sex as a biological variable, SABV is the term you may know, will be factored into research designs. And in fact, now you really need strong justification in order to only use one sex in your studies. So adequate consideration of both sexes in experiments touches on all of the 3 Rs, specifically refinement of the study design themselves, replacement of solely males, and reduction to ensure that studies have the correct number and both sexes of animals needed for a statistically relevant outcome. So I just want to state again, as a reminder from Russell and Birch's original work and from discussions with my colleague, Dr. Taylor Bennett, who put out a paper in 2015 on the 3 Rs, that the 3 Rs principles originally did not define reduction as minimization of the number of animals used. In fact, Russell and Birch emphasized that it's often impossible to know in advance of an experiment whether the animal numbers will be used and if more than what the minimum number is going to be. They really just took the minimization piece out of it and emphasized statistics and analysis to make sure that the method is appropriate for the true number of animals needed. They were advocating, as a reminder, for the new science of a humane experimental technique. So aiming at absolute minimization is really illogical in the face of experimental design. Animals will be wasted if too few are used, and results won't be able to be interpreted or repeated. So please just keep in mind that the term reduction intersects with refinement. We also think about reduction of pain, distress, and stress. All of these demonstrating a more progressive and expansive 3 Rs interpretation. So another approach to considering harm-benefit analysis of animal experiments was presented in 2017. And this was by Hanna Verbal. And the author here stated that if studies don't produce results that are scientifically valid and reproducible, then animals again will be wasted because it's inconclusive research. It doesn't matter how little harm or how much harm is inflicted on the animals themselves will not lead to reproducible results. So what I'd like to just point out quickly, and I encourage you to look at this paper if you've not seen it, is that he mentions validity. So the 3 V's for construct validity, internal validity, and external validity. So what the author here really emphasizes is that it's a 3 R's effort to minimize animal use, which again is this somewhat constrained, antiquated idea of reduction that may inadvertently exacerbate limited data output. So what his concept is saying is that we can't just have each individual lab doing the exact same experiments and getting different results. We need to have more discussion from multiple laboratories, repetition of experiments to try to produce generalized and reproducible outcomes. And then the final thing that I wanted to mention, which is so recent and has been really well done, is the 6 Moral Principles of Animal Research and Ethics, which came out in a book by 2 preeminent ethicists, Tom Beecham and David DeGrazia, just last year. And it's really nice. It breaks things down. So it's a slim text. Chapters are authored by well-known professionals that share these stakeholder perspectives from veterinary medicine, biomedical research, physiology, psychology, law, and bioethics. And it offers a framework that bridges the gap between concerns of the biomedical and the animal protection communities. So I wanted to thank again Dr. Bennett for alerting me to an ALAS presentation from last fall that had Mimi Riley and Dr. Margaret Landy presenting on how to apply this new ethical framework. They looked at current case studies and applied the 6 principles. So the way this is designed are 3 principles of social benefit to humans versus 3 principles of animal welfare. And as they, both my colleagues pointed out in this ALAS talk, these 6 principles were not made to compete or replace the 3 R's, but they are an important next step as we continue to expand and explore the path for holistic assessment of how we use animals in research. They do tend to reflect a little bit more on the classic 3 R's, those bullet point definitions, but they, I think what I'd like to prove to you all is that the 3 R's are much more dynamic in how we interpret them. So in fact, instead of contemporary 3 R's, I want to present a progressive 3 R's approach to think about all the concepts and encompass them all entirely. So I'm really thinking more now in our umbrella analogy that this is a symbol. It's not really just an acronym. Stakeholders really need to be having group discussions about the 3 R's that rigorously evaluate science. And this is, and the umbrella model allows us to encompass the ongoing discussions that came out in the government principles, in the 4 C's, when we look at the 3 V's, and now most recently with the 6 benefits. So as an acronym in a progressive way, it covers and includes successive and related concepts. And there will be many more concepts still to come out on this topic that we can roll into the umbrella of the 3 R's. And again, importantly, we just need to keep discussing this to benefit, to the harm benefit, the ethics and benefit analysis. Again, as I mentioned, I'm not the first person to be talking about these things. I really encourage people to look at the NC3R site. So what they have defined as standard is what I call classic in our definitions, and what they call contemporary are the progressive definitions. So earlier when we look at when we were talking about, reduction equaling minimization, that's actually not really true. Reduction is much more robust in our definition, how we can apply it to all the speakers that are presenting today and all of the projects that we'll be talking about in the future. So I want to bring just back to art here, the changing landscape of animal research. Certainly the last year of the pandemic has resulted in a massive shift in how the world views science, discovery, and animal use, and we are in a changed world. So in the art world, the landscape painting is based on the use of color theory. Different colors are next to each other and it alters the eye's perception. So we look at this, the strokes are very expressive. They take on a physical presence of their own. This is Van Gogh's wheat field. And if we stand back and look at it, it makes sense. It is a wheat field, but there's so much more to it when you lean in and assess the colors throughout. It's the individual strokes that make the difference of this whole painting. So we really want to be thinking more broadly about what's to come in the new world. In particular, the pet proliferation. There's too many, pandemic pet proliferation, my alliteration here. So I did not get any new pets, but I would be interested to hear how many of you decided during the pandemic that you would like to add a new furry member to your household. So we've seen this proliferation. This is an article that came out in the Michigan Vet Med Association about the increasing bond between human and animals. And research has even been conducted in this area as well about when we've had so much social distancing, social isolation, that having pets involved and the many different types of pets has really been very, very helpful. So this in and of itself, working with animals, has explained a lot about how we will be thinking about animals for years to come. And certainly not in the terms of reduction of animal numbers, but in refinements and reductions of stress for the benefit of humans, this is applicable. And if we just think about all of the different species here, some of these are pets, but all of them impart somewhat of an emotional, often a physical response when we view them, depending on our perspective. And we're still learning as a society about all of these species, about which opportunities can be provided to assist them with expression of species-specific behaviors, about how they may contribute to our societies. One of the things that we've learned through the literature in the lab animal field is that the provision of enrichment just by itself, even in the absence of drug treatments, can modify pain behaviors and diminish pain more rapidly in mice and rats. So we can also better assess social housing for social species, which is a main area of focus for welfare. It's a key technique, but again, requires some judicious oversight because some social species can fight with each other, cause injury when they're establishing dominance. So I bring up these points just to continue to rebuff this sort of societal pressure, sometimes from the media, to just reduce animal numbers and fully replace all animals from study, because we study animals for the sake of animals as well. And it's a key consideration in many research proposals. These are goals enmeshed in the expansive and future 3Rs concept. So we've talked a little bit about animal models. These help us to understand the pathogenesis and mechanisms, certainly in the pandemic for SARS-CoV-2 disease and biology. As I mentioned, others have already commented on our challenge with accessing non-human primates, given past pressures to decrease availability and use. And as a reminder, it's the science itself that's going to drive our ability to reduce animal numbers in certain disciplines. For example, using former animal modeling data to accelerate the COVID vaccine development. Some would argue that we have an ethical obligation now more than ever to drive science forward and create new knowledge, which I'll remind you goes back to the US government principle 2. Procedures should consider relevance to human or animal health, advancement of knowledge, and the good of society. And I was glad to see that Dr. Bohm presented earlier on the active group. This is from NIH. A paper was put out that I encourage you to take a look at. It just came out recently at the end of 2020. NIH, as a reminder, established this partnership in April of last year to bring together experts in the fields of virology and public health, vaccine, and drug development. And I'd just like to quote from the paper, under more normal circumstances, the response to emerging pathogens is a methodical linear progression of in vitro, then in vivo, and then clinical studies. Yet the urgency of the response to the current pandemic required these activities to be carried out in parallel. It was prior experience, including robust animal studies with other coronaviruses, that provided the guiding foundations for current research, allowed for the rapid vaccine development and the application of interventions to the disease. So I just really feel we have so much more to learn. Groups like this are going to be really inspiring, and partnering across all of these disciplines will really better prepare us to deal with future crises. So I was asked in the last few slides to just touch on another initiative that NIH has been actively involved with over the last several months. They established the Advisory Committee to the Director, or ACD, working group. And this group was charged with assessing and making recommendations to enhance the reproducibility and rigor of animal research. So I was asked to join this committee. We had our kickoff meeting and address from Dr. Collins in September of 2019, and we were able to have one in-person meeting at the NIH before the pandemic shutdown began. However, undeterred since that time throughout 2020 and as recently as last week, we have been meeting almost monthly and more frequently now as we come to the end of our charge to continue to address the goals and divide into subcommittees to address specific topics. And just so everyone's aware, next week on Friday, the final report is scheduled to be delivered to the NIH. The makeup of the committee is here. These are the external members outside of the NIH. It was a broad swath of scientists, journal editors, experts in a variety of animal models and species. And as I mentioned, I was on this as well as another veterinarian, Dr. Brian Berridge, who is on the next slide. These were the internal members. Brian is a boarded pathologist as well. So it was a really interesting complement, very robust discussions. I certainly learned a great deal by being part of this group. So the main charge to the working group are itemized here on this slide. We had to look at gaps and opportunities for improvements. We really had to think about how animal models of human disease are developed and validated. Think about alternatives as well. There was discussion about registering animal studies, to again have another level of review. Thinking about the financial implications of these potential changes is a big thing for how the scientists may be impacted. And then also talk more broadly about how rigor will be incorporated into scientific training. I mentioned that there were subcommittees involved with this. My own was involved in addressing the extrinsic factors that are inherent to working with animals. This is a subject that's near and dear to my heart. So for those who maybe wouldn't know how to define that exactly, there will be glossary that's included in the final report. And extrinsic factors are defined as housing, husbandry, handling, feed, water, bedding, enrichment, pretty much anything, light cycles, et cetera, that would have a direct impact on the experience of the research animal during that course of experimental phase. So obtaining reproducible results in animal studies requires our research teams to undertake a critical evaluation of all aspects of the research design, including the impact on and experiences of the animals. So the goal for reproducibility would include steps necessary for researchers to report on critical identifying information from the grant proposal to the final published manuscript, including things like really applying Arrive 2.0 and making sure that the details are present. So the final report is going to have multiple themes related to study design, research practices, selection of animal models, the reporting mechanisms, and how to measure effectiveness of what the group has come up with related to improving rigor, reproducibility, and translatability. And then just in conclusion, I am hopeful, and my goal today was to convince you to advocate for the continued evolution of the 3 Rs. As partners within our community, we have an amazing number of advocacy organizations for animal medicine, science, and research. I'll list them on my next slide. Their members include scientists, technicians, husbandry staff members, supervisors, the public, disease survivors, regulatory administrators, and lobbyists. Altogether, we create momentum that can become momentous. We are a veritable chorus of voices that assert ethical responsibility to enhance reproducibility, to ensure welfare practices, to support the responsible use of animals, and to invest the necessary resources toward valuable outcomes for animal and human health. So in conclusion today, reconsidering the classic 3 Rs with progressive interpretations in combination with many other ethical tools of assessment is going to be essential in the ongoing intricate considerations of appropriate and future animal use. I thank you very much for your time. I really want to thank Dr. Brown for the invitation today, and here are the advocacy groups, many members of whom I've talked with and learned from and will continue to do so in the future. So thank you so much.